Hello, my name is Chang Lim. I would like to present about developability assessment and high concentration formulation platform today. First of all, I need to share about our client molecular trend. For this presentation, I reviewed previous client molecular complexity and concentration trend. As you can see, the molecular complexity was increased. On 2022, 60% of our project were simple molecular antibodies. But now, most 60% of molecules are a complex protein like fusion, multispecific, or recombinant proteins. Concentration perspective, over 100 mg per ml concentration expectations were increased from 18% to 24%. Molecular complexity is okay, because many of our clients want to enhance their molecular capability using T-cell engager or multiple capture using one molecule. But why? Why also our client wants to get high concentration formulation for their molecule? I thought two reasons why our client wants to high concentration formulation condition. First reason is the trend of ready to use and easy to use. Most of patients want to inject by themselves at home. They don't want to go clinic for IV infusion administration anymore. To meet their expectation, since only 1.5 or 2 ml is possible using sub-Q, the protein concentration must be increased compared to IV infusion. So, high concentration formulation also opened the door to further options, such as self-administration therapies, which lead to improved convenience and compliance. And the other reason why they want to get the high concentration, because high concentration formulation also save the cost in terms of storage and shipping, as the high concentration drug product requires lower volume. Hence, smaller vials or syringes are required compared to lower concentration drugs. But high concentration, like over 100 mg per ml, is not easy due to those challenges like pH shift issue due to Donan effect and instability issue during the thermal or physical stress study. For example, aggregation, formation, haze appearance, visual particle issues. These issues can be occurred more at high concentration because of the solubility and hydrophobicity. And also the high viscosity issue at a high concentration can be occurred. If the viscosity value exceeds 20 CPS, then the DP cannot be injected using general syringes or preferred syringes. These issues generally occur due to molecular characteristics, and to solve the problem, then let's think, can these issues be solved for high concentration? No, mostly we can. Do you know the word, if you know the enemy and yourself, then you never lose? Much protein formulation experience is required to solve those problems because most of issues can be solved by the scientist's background knowledge. That's why we launched SICON, High Concentration Formulation Platform in Samsung Biologics. Our SICON platform can confirm those issues before starting the formulation development and prepare a strategy for the issues occurring, and finally achieve the high concentration formulation condition, which has good stability, low viscosity at over 200 mg per ml. This is SICON workflow. All steps are important, but most important thing is gate check step. At this gate check step, we confirm those potential issues that I already mentioned, like pH shift, viscosity, and stability, especially aggregation. If the molecule's high concentration is possible, then just go forward. But if the molecular high concentration is not possible, then we prepare some strategies to solve the problem using other set of excipients like viscosity reducers, stability enhancers used during TFF process, etc. Let's see one by one. This case is a molecular antibody case, and the target concentration was 210 mg per ml. But to get the manufacturability, we concentrate that to 266 mg per ml. At the 266 mg per ml, the molecular pH condition was shifted to 0.1. We confirmed this during gate check step. So we can prepare the offset buffer, and finally, we met the optimal pH condition using the offset buffer. We also confirmed not only viscosity, pH shift, but also confirmed the whole time stability, because sometimes DSP process is not complete within a day. We confirmed the whole time stability during 3 days at ambient condition. You can see in this case, for 266 mg per ml, the molecular stability was not changed during 3 days at ambient condition. This step was performed after pH buffer selection, so we knew pH buffer was selected through this result. I also would like to say about the importance of excipient selection. Please look at the left side. To bio, there are same molecule with other excipient set. Left is clear, but right is not clear. Why this issue happen? Left side bio was used non-electrolyte excipient, and right was used electrolyte. As you see, electrolyte excipient is not matched to this molecule. Also, you can see right side graph. Different excipient shows different stability result after thermal treatment. That's why I emphasize the scientist's background knowledge is important to choose the right excipient type and condition for the molecule. I also said about the viscosity. Let's say the molecular stability is okay, but the viscosity result is over 20 CPs. Then how do you think? Is the molecule can go to the clinic? I think no. I don't think so. The viscosity has to be reduced below 20 CPs. As you see, over 200 mg per ml case, we can manage the viscosity below 20 CPs. As per this testing, we get 15 CPs for the 200 mg per ml using SICON formulation screening platform. So our SICON platform can achieve over 200 mg per ml with well-managed low viscosity and good stability. To get the high concentration like this, already said, knowing the molecular characteristics is very, very, very important. Actually, not only for high concentration, but also overall drug development process, knowing the molecular characteristics is important to avoid issue occurring. Let's say one molecule was discovered by discovery scientist and went to CMC development, but unfortunately, some issues occurred. The program was stopped. Honestly, many molecules cannot pass this step. Then how much time and money was spent? Can you imagine that? For a small biotech, that is really disaster. So know the molecular characteristics before starting the CMC development is very important to save our client budget and time. That's also why we launched our developability prediction program, Developability. Developability can detect the potential issue using this workflow. At first, we perform in silico-based molecular primary structure and perform relative solubility, compound basic physical chemical characteristics, certain thermal stability confirm, and low and high pH stress stability confirm, and hydrophobicity confirm. We only use 80 mg of protein within a month to confirm these all data to prepare GMC development. Since 2020, when we have launched the Developability, this program has been improved to version 3. At first, we only have basic characteristics and thermal stability and low pH confirmed. But now, we have solubility testing, hydrophobicity testing, and in silico assessment function. Let me introduce our in silico molecular assessment function. Since this is using primary structure, I cannot say this prediction is 100% accurate. However, I can say this molecular assessment can detect several hotspots like oxidation, deamidation, aggregation, or cleavage on the molecular primary structure, and using this data with wet work data, we can evaluate the molecular characteristics during Developability. Thanks to AI&I team in Samsung Biologics, we can have our own sequence analysis program. We can analyze our client sequence to get those information without uploading to website. So that means our client does not need to open their sequence information to web environment. This is an example of this program operation. I analyze it to sequence which molecule is already commercialized one. As you see, our own program can analyze all basic information like PI, EC value, stability prediction parameter, and also sequence information. We also can evaluate molecule like this. This is fusion protein case. We can compare two regions of this molecule. As per data, VH region is a little bit higher risk for oxidation and deamidation than VL region. Using this data, we can prepare our formulation strategy to mitigate the risk occurring. And the project was succeeded. We also can detect all cleavage sites of the sequence using our program. This information is based upon previous well-known enzyme cleavage site database. So our Developability can detect all cleavage sites using this program. As I mentioned, we confirm T-onset TMT aggregation using DSF SLS system. We can compare those data for each candidate to know the thermal stability characteristics. And we also perform DLS analysis to confirm the polydispersity and hydrodynamic size for the target molecule. We also can perform B22 KD measurement to confirm video coefficient and diffusion interaction parameter. And also, we can perform delta G measurement to confirm the chemical stability. These are cases we analyze for KD. Please look at the left upper side graph. If the molecule has positive KD trend, then the molecule has repulse characteristics each other. If the molecule has negative KD trend, then the molecule has attractive characteristics each other. That means this molecule has more chance to aggregate. Using this, we can know what the molecular characteristic. These molecular results are correlated with SEC result data after thermal stability. After 2 weeks thermal stress treatment, aggregation and fragmentation will be confirmed using SEC analysis. Using this result, we prepare specific strategy for the formulation development of the target molecule. I'd like to say about relative solubility study. Since this stage, not much material is supplied for developing prediction. To confirm solubility using small amount material, we use PEG solubility method. Using this method, we can predict the molecular solubility through comparing previous our database. Let's see the PEG solubility result. upper graph, you can see various pH solubility graph for various molecules. Through this comparing, we can predict the target molecular stability. And this database is accumulated for various types of molecules to compare the protein of interest. Honestly, relative solubility test results accuracy is not 100%. So if our client wants to confirm the real solubility, then we will do real solubility study using TFF system. As you see, we concentrate using TFF system to the target concentration with viscosity monitoring. And we also will provide a whole time stability study result to confirm the feasibility of DSP process. But this testing needs much protein amount to use. We also perform hydrophobicity testing. Hydrophobicity characteristics is directly linked to the solubility and molecular stability, especially aggregation formation. So knowing the hydrophobic characteristics is very important to predict the feasibility of process development. For this evaluation, we use HIC analysis method, and we have specific this HIC result database to compare the target molecule. So using Developig, we can evaluate the molecular characteristics using various tools to know the molecular process development feasibility using only 80 mix within a month. Now we have Developig 3.0. Using this, we can offer the advantage of facilitating proactive preparation for process development through the hydrophobic testing. And this Developig platform is continuously being improved with significant enhancement, particularly for the in silico aspect. Thank you. Now I'm happy to take any questions you may have. That's actually a question I received before at the speaking session I attended. We don't use DLS to measure viscosity. We use rock and honeyburn system, which required only 5 to 450 microliter of sample. So we can measure viscosity using mid-throughput or high-throughput system instead of DLS. And there is no concern about the protein amount. Thank you.